Have you ever heard of a drug called 𝗤𝗚-𝗟𝗏? No? Well, it's a rescue drug that rose from the ashes when an earlier version of itself failed in the clinic. Let's have a look. This story begins with a very promising target, the interaction between PD-1. Bristol Myers Squibb, or BMS, in collaboration with PeptiDream Inc., set out to develop small molecule inhibitors of the PD-1 pathway. They started off by creating macrocyclic peptide inhibitors, which are basically cyclic molecules that mimic the natural ligand and bind to the receptor. They found that these compounds showed potent anti-tumor activity in vitro, meaning they could effectively block the interaction between PD-1 and PD-L1 in a Petri dish. This looked very promising. However, when they tested these compounds in clinical trials, they hit a wall. It turned out that these molecules were rapidly cleared from the body, resulting in suboptimal half-lives and limited therapeutic efficacy. This is a common problem with many new drugs. Sometimes their structure makes them get quickly broken down by the body, which limits how much of the drug can actually make it to the site of action. But the scientists weren't ready to give up, so they went back to the drawing board. They wanted to find a way to improve the pharmacokinetic profile of their lead compound, 𝗤𝗚-𝗟𝗏𝗡𝗚. In other words, they needed to find a way to make it stay in the body longer. To achieve this, they engineered a second-generation inhibitor, 𝗤𝗚-𝗟𝗏𝗡𝗚, which incorporates a human serum albumin binding motif. Human serum albumin, or HSA, is a protein that's abundant in blood plasma. It's responsible for transporting various molecules throughout the body. The binding motif is like a little hook that allows the drug to latch onto HSA, and this interaction is reversible, meaning the drug can come off when it needs to, but it significantly prolongs the drug's half-life by preventing it from getting lost in the body. Think of it like this. If the drug is a little boat and HSA is a life raft, then the binding motif is like a rope that allows the boat to be pulled back from the water when it starts to sink. In this way, 𝗤𝗚-𝗟𝗏-𝗡𝗚 can hitch a ride on the long-lived HSA, which has a half-life of about 20 days, and stay in the body longer than its predecessor. This clever strategy has paid off. In preclinical studies, 𝗤𝗚-𝗟𝗏 showed a favorable pharmacokinetic and pharmacodynamic profile, suggesting that it can reach therapeutic concentrations more easily and effectively block PD-1, PD-L1 interactions. So from a failed clinical trial to a potential game changer, the development of 𝗤𝗚-𝗟𝗏 shows that sometimes all it takes is a little creativity and a lot of perseverance to turn things around. If you like this video, check out our video on how the first cancer immunotherapy drug was discovered. And for more videos of us uncovering the science behind the headlines, make sure to subscribe.